So in this session, I'm going to talk about eukaryotic genome as it exists in other organelles than nucleus. And what are the other organelles I have been talking about? That is the chloroplast and mitochondria. So how the organellar genome organizes itself in these organelles? What are the differences between nuclear genome and organellar genome? How it is different, and how the inheritance patterns differ from nuclear to organellar genome. So these are the aspects which I will be discussing with you in this session. So when we talk about organellar genome, I'm referring to these two organelles I mentioned, that is chloroplast and mitochondria. Historically, if we see, the presence of DNA was shown as early as in 1960s, and in fact, around the same time, both discoveries were made about chloroplast and mitochondria, that both of them have the genetic material, that is DNA. And if you look at structurally, the nucleoid kind of structures were seen both in mitochondria and chloroplast, which had the DNA in them. Okay. Now, if we see what are the features of, or salient features of organellar DNA, or in today's term we call organellar genome. So, like nuclear DNA, chloroplast and mitochondrial DNA also replicates, and it follows the same process of semi-conservative mode of DNA replication. So there is no difference in that. And like wherever DNA is, it is liable for mutation. So like nuclear DNA is liable for mutation, so also organellar DNA is also liable for mutation. But then they are expressed and inherited differently from nuclear genome. We will see that subsequently or later on. And the DNA, the organellar DNA, is transcribed and translated in the organelles only, whereas if you see the nuclear DNA, it transcribes in nucleus and translates in cytoplasm. Okay, and both mitochondria or chloroplast DNA, they show uniparental inheritance. This is different from what we see for nuclear DNA, which shows biparental inheritance. Okay, and then organellar DNA present in multiple copies, actually. Like for example, chloroplast has 20 to 40 copies per chloroplast, and so mitochondria has 4 copies in yeast, for example. So as against nuclear DNA, the organellar DNA is present in multiple copies. When I say multiple copies, it is the same DNA which makes its own copies and is present in the organelle, like chloroplast and mitochondria. Now, if we look at that, how these two organelles originated in course of evolution. So, an endosymbiotic theory has been proposed that says that the archaebacterial cells is the first cell type which gets originated, and then protozoans and the cyanobacteria. These are the simple unicellular forms which came into existence. And as the time passed by, the protozoans got into the archaebacterial cells and later on got differentiated into mitochondria. And when this cell, archaebacteria and protozoan combination got fused with cyanobacterial cell in evolution, it gave rise subsequently to the plant cell or plant kingdom. So that's how eukaryotes, like plants and animals, got evolved. So animals have mitochondria in it, and plant cell has both mitochondria and the chloroplast. So origin of chloroplast is a cyanobacterial cell, and mitochondria is from protozoan protozoan forms, or what is called proteobacteria. You know? Now, what supports this theory of endosymbiotic theory? That is, there are certain features which support this. That is, both mitochondria and plastids, which are which is the initial form of chloroplast, they both are formed by a simple process of binary fission, which is seen in bacteria. That's how bacteria divides itself just by fission. So both mitochondria and chloroplast, they um, they they are formed by binary fission. Okay, and then the DNA, both in chloroplast and mitochondria, is very much similar to the bacteria, in size, and also it is circular in nature. Okay, so it resembles with that of bacteria, and both these organelles, mitochondria and chloroplast, has ribosomes, like 70S ribosomes, which is the characteristic of bacterial cell for protein synthesis. Okay, and the mitochondrial DNA codes for many enzymes, similar to that of bacterial cells. Okay, so what I'm trying to say is that the features which the organellar DNA shows, both in its structure and mode of replication, it resembles with that of bacteria or prokaryotes, in other words. Now, what are the differences between organellar DNA and the nuclear DNA? So, first difference is the presence. Organellar DNA found in chloroplast and mitochondria, and the nuclear DNA is found in the nucleus. The organellar DNA is circular in nature, except in protozoa, ciliate protozoa, where it is linear, whereas the nuclear DNA is linear and present in chromosomes. And very important difference between the two is the synthesis. The synthesis of DNA continues through the cell cycle, throughout the cell cycle, whereas in nucleus, the DNA synthesis during cell division is restricted to the synthetic phase of the S phase. Okay. So this is the difference in the two forms of DNA in organelles and in nucleus. Now, if we see how these three DNA interacts or crosstalk between in the cell. So in cell, you have nucleus, mitochondria, and in plants, you have nucleus, mitochondria, and chloroplast. So if you look at this diagram, you have, see, environmental signals are basically perceived by all three. Nucleus perceives the signals, chloroplast, as well as the mitochondria. And in turn, these two organelles send signal to nucleus, and nucleus in turn it perceives the environmental signals and sends signals to chloroplast and mitochondria. And both these organelles also crosstalk among themselves. So basically, both the DNA, all the three types of DNA, nuclear, chloroplast, and mitochondria, they coordinatedly respond to environment, basically. This is what it's trying to put in here. Now, focusing on to the mitochondrial genome. So, what happens? What are the characteristics of the mitochondrial genome? You have it is circular or linear, as I told you, in case of protozoans, and but by and large, it is circular in nature, except one exception. The size also varies from lower to higher eukaryotes, from 16.5 kb to 100 kb. Okay, and this kind of variation is seen more in mitochondrial DNA as compared to chloroplast DNA. So what I'm trying to say is the mitochondrial DNA or the genome shows more variation in their size as compared to chloroplast genome. And in higher plants, the mitochondrial DNA shows the homologous sequences, and it shows recombination within themselves, which generates subgenomic circles with full genome. So, in other words, plant mitochondrial DNA shows a lot more variation of this kind as compared to animal mitochondrial DNA. And in mitochondrial DNA, the genes are located on two strands, heavy strands and the lighter strands. Heavy strand has more genes as compared to the lighter strands, and the mitochondrial DNA codes for rRNA, tRNA, and the proteins which are required for respiration, basically. But they do not code for any structural protein. Structural protein means the one which participate in structural organization. So all these proteins which are coded by mitochondrial genome, they participate in the functions of the mitochondria. And it is present in the mitochondrial genome is present in the nucleoid region of the mitochondria. And as I mentioned, the number of mitochondrial genome varies from organism to organism. For example, you have in yeast 4 to 5 mitochondrial DNA molecules per nucleoid, and each mitochondria has 10 to 30 nucleoids. So you can imagine that there are 4 to 5 molecules per nucleoid, and each mitochondria has 10 to 30 nucleoids. So you can multiply that how many DNA molecules will be there per mitochondria. For example, if we look at the human mitochondrial genome, It contains no introns. Mitochondrial genes, they do not have introns. Now, this is the striking difference between nuclear gene and the mitochondrial gene. Nuclear genes have, they organize themselves as introns and exons, whereas the mitochondrial genome or the genes, they do not have introns. But introns are seen in other organelle genes like chloroplast genes they have. And the human mitochondrial DNA codes for tRNA, rRNA, and the proteins like cytochrome C and cytochrome oxidase, NADH dehydrogenase, and ATPase. These are the functional proteins which are they participate in the respiration process. Now, if we see the diversity of the mitochondrial genome, so if you see the eukaryotes, if you put in animals, fungi, and plants, okay, so if you see the size of the mitochondrial genome or the genome of these three, in the three types of eukaryotes like animals, plants, and in between you have fungi. So in animals, the size of the genome varies from 14 kb to 42 kb, whereas in plants, mitochondria 184 kb to 2004 kb. So that's the variation in the mitochondrial genome size. And in between you have fungi where it varies between 17 to 180 kb. Now, non-coding DNA in the genome, in animals, it's very low. Means most of the mitochondrial genome codes for something or the other. It has very, very low amount of non-coding DNA. And I discussed, if you recall, in the nuclear genome, you have large portion which is non-coding, and the coding part represents 2 to 10%, Okay. Now, coming back to non-coding part in the plant's mitochondrial genome, it's pretty high, and it is variable in fungi. So animals have very low non-coding region, whereas in plants, it's pretty high. Mutation rate is high in animals and very high, very low in plants. Okay, recombinations absent in animal mitochondrial genome and is seen in plant mitochondrial genome. Introns is seen only in plant mitochondrial genome genes. Okay, and it follows. The plant mitochondrial genome follows the universal genetic code, whereas animal it does not. Okay, and in fungi mostly they follow. So this is the sort of differences between the mitochondrial genome of animal and the plants. So there is distinct differences between the two genomes of mitochondria in animals and plants with respect to their size and behavior. And this is just to show that all the different kinds of mitochondrial genome or genome from different sort of eukaryotic organisms like animals, plants, fungi, including humans. These genomes have been sequenced, annotated, and now we know what these genomes contain, what kind of genes they contain, and how these genes help in performing the cellular functions. Okay, so I stop this discussion here on mitochondrial genome, and in my next video, I will talk about the chloroplast genome.